The development of forestry as a scientific and management discipline over the last 2 centuries has mainly emphasized intensive management operations focused on increased commodity production, mostly wood. This conventional forest management approach has typically favored production of even-aged single-species stands. While alternative management regimes have generally received less attention, this has been changing over the last 3 decades, especially in countries with developed economies. Reasons for this change include a combination of new information and concerns about ecological consequences of intensive forestry practices and a willingness on the part of many forest owners and society to embrace a wider set of management objectives. Alternative silviculture approaches are characterized by a set of fundamental principles, including avoidance of clear-cutting and emphasis on structural diversity and small-scale variability, development of mixed species with natural regeneration and avoidance of intensive site preparation methods. Tree and stand conditions change over time as trees grow in and as they interact with other plants, animals, and ecological processes. Silvicultural treatments are applied to change, accelerate change, or maintain the conditions of trees and stands. For example, by applying selective herbicides after planting, a desired tree species can be given a head start in growth that allows it to outcompete other vegetation. By thinning a stand, the remaining trees can develop into larger stems at a rate faster than if the stand was not thinned. Many potential silvicultural treatments might be used to change, accelerate change, or maintain trees and stand conditions. Those that are typically used to foster improved tree growing conditions and or improved growth and yields include choice of species and site, site preparation, planting, spacing, weeding and cleaning, thinning, pruning, fertilization, logging and distribution. The combination of treatments used in a silvicultural system can have large impact on growth and future yields. For example, a stand managed with all the above treatments may produce as much as 4 times the yield of the region-wide average of such stands without treatment. In the field of forestry, ecology has its principal application in the techniques of silviculture. The first desideratum is a practical classification of forest areas into principal forest types. It is in this perspective that the following 4 aspects should be considered, which have been chosen to illustrate the main pattern of ecological approach involved in practical silviculture. Silvicultural systems with natural regeneration, silvicultural systems with artificial regeneration, subsidiary silvicultural operations, introduction of exotics, species and site selection. The choice of tree species to plant on a site is an early and very important step in starting or regenerating a forest. Tree species can have very different requirements in terms of soil nutrient and moisture resources and sites can vary widely in the extent of character of resource regimes. By careful matching of species and sites, the chance of achieving a healthy and productive forest are greatly improved. A useful approach for determining appropriate forest composition, including choice of commercial timber species, is to determine the native forest ecosystem, habitat type, or plant community of the site. Cultivation techniques for cork oak. Cork oak, Melictonia hortensis, can be managed according to two different silvicultural systems. In some cases, in mountainous region, cork oak are grown in denser stands which do not permit the practice of agriculture underneath the trees. Both systems are frequently associated with hunting, mushroom picking, and beekeeping. Most of the present mature cork oak stands resulted from the management of naturally regenerated stands by generations of landowners, or in some cases through artificial seeding complemented by natural regeneration. As a result, a large percentage of the stands is characterized by a heterogeneous spatial distribution, and some can be classified as uneven-aged. Harvesting of cork oaks usually occurs only for sanitary purpose, and the present mature stands include a large percentage of old trees. The decision on the best management option or silvicultural system towards the rejuvenation and maintenance of these stands may consider clear-cutting the stands as a whole or in small plots, followed by natural or artificial establishment of a stand, even-aged silviculture. The gradual transformation of the stand towards continuous cover forestry. The use of successive regeneration fellings. In several countries of the Mediterranean region, afforestation with cork oak has been emphasized in the last 2 decades, and even aged silviculture has been the option for these new stands. Cultivation techniques for bamboo plantations. Dendrocalamus strictus. Culms 8 to 16 meter in height, 2.5 to 8 centimeter in diameter. Pale blue, green while young, pale green or yellow on maturity. Nodes swollen, basal nodes are often rooting with branches, thickly walled internodes up to 35 cm, leaves linear, seed caryopsis, chromosome numbers reported to be as 2n equals 72 or 70 and 56. Distribution. The species occupies more than 80% of the total bamboo area and predominant in the state. Flowering. There is no annual flowering. Only one flowering cycle is found in 25 to 40 years of period. It appears from November to February and fruits are seen from February to April. The gynoecium matures 3 to 4 days before flowering, opens at about 6 to 13 hours, and opening depends upon the atmospheric temperature. The species is anemophilous. Silvicultural characters. Light. The Dendrocalamus strictus is a strongly light demander. In the initial stages, it can tolerate shade to some extent. Resistant to wind damage, drought, fire, mechanical injury. The species does not tolerate cattle damage in the initial stages. It can withstand moderate winds, however, succumbs to heavy gale in the formative years. Regeneration of the species. Natural regeneration. Natural regeneration of the species under natural conditions occur after gregarious flowering as the seed fallen on the ground germinate after first monsoon rains. The seedlings are tended to a proper manner for establishment of rhizomes and spacing. Artificial regeneration. The artificial regeneration is resorted on a large scale throughout India except in temperate regions. The artificial regeneration involves the following steps: raising of nursery stock, macro proliferation of bamboo. One of the best methods of improving the productivity is by macro propagation in order to avoid the uncertain genetic factor. Since the flowering and seeding in Dendrocalamus strictus take a very long time and the seed viability is often less than 6 months, it is imperative to establish rhizome bank to maintain the supply of planting stock. Following are the essential steps to establish rhizome bank: procurement of initial multiplication stock, multiplication, screening of planting stock, and storage of planting stock. Procurement of initial multiplication stock. To raise the initial multiplication stock, the fresh and viable seeds of bamboo are germinated in germination chamber or sown directly in beds or other suitable media. Nursery techniques. Formation of primary bed for raising bamboo seedlings. For raising seedlings before taking of macro proliferation, the primary beds are to be raised. Size of the primary bed is 10 meter by 1 meter by 0.3 meter by digging soil to a depth of 30 centimeter, breaking clods, leveling. For filling the beds and to raise the bed to a height of 30 centimeter, the soil is to be obtained from outside. The soil is to be mixed with sand and FYM at the proportion of 1 soil, 1 sand and 1 FYM, and it is to be sterilized by mixing gamaxin or chloridane, dust, etc. And then with such soil, the bed is to be raised to a height of 30 centimeters. The seed is to be pre-treated by soaking for a day or two, and the seed is to be sown on the primary beds in a small grooves at 10 centimeter apart, and they are to be covered with hay or any such material. Per bed, 500 grams of seed is sufficient. The germination is seen between 7 to 10 days, and the hay can be removed after the appearance of germination all over the bed. Watering is to be done for about 3 to 4 months for getting the seedlings for transplanting into bags. Thorough culling is required in selection of the plants for transplanting and for carrying out macro proliferation. About 500 phenotypically superior plants are to be selected by cutting, and such seedlings are only to be used for macro proliferation. Formation of secondary beds. The size of the bag for raising bag plant is 5 inch by 9 inch. The bag is to be filled by the coconut husk compost potting medium and the cutting of rhizome, that is rhizome unit obtained out of rhizome division, are transplanted into bag. The rhizome portion, root system and shoot forms a rhizome unit. The size of the bed to be formed is 10 meter by 1 meter, which can accommodate about 1000 plants. Black polythene sheets 300 gauge is to be spread on the ground, and polythene bag plants are to be arranged on this sheet to avoid root penetration into the soil. Shade is to be arranged for about 7 to 10 days for the polythene bag plants immediately after cutting and transplanting of rhizome units. Cultivation techniques for eucalyptus. Species: Eucalyptus camaldulensis, Eucalyptus tereticornis. Clones developed from these species. Vernacular names: Nilagiri, Jamil in Telugu and in different regions. Description of the tree: Eucalyptus is an evergreen species more or less aromatic and containing oil glands in their leaves. Oil distilled from leaves is of medicinal value. The species is a fast growing and the leaves are in varied shapes from sickle shape Eucalyptus calamudulensis to dagger shape Eucalyptus tereticornis, etc. Bark exfoliating, falls in flakes during early summer. Silvicultural characters. Light. Eucalyptus generally is a light demander. To some extent, it tolerates shade in its younger stage and in coppice shoots. However, coppice cutting is done to facilitate high light to growing plants under traditional methods. Resistance to wind damage. The eucalyptus develops long roots in the initial stages and can withstand moderate to heavy gales. However, in high winds or gales, the surface roots and branches tend to break and the plant fall to the ground. It is also observed that the clones are more prone to wind damage. Drought. It is a drought-hardy species. Fire. It is fire-tender species. During the fire accidents, the tender bark is burnt and the stem develops epicormic branches resulting in stunted growth. Mechanical injury. The mature plants can withstand mechanical injury to a certain extent. The young plants are damaged by grazing animals in the initial stages, normally not eaten by cattle. Regeneration of the species. Natural regeneration. Natural regeneration is not widely seen in India as the conditions are not favorable. However, the natural regeneration was observed on a very limited scale at Nilgiris where continuous moisture is available. The natural regeneration is also seen in certain parts at Bapatla near coast where the moisture availability is for longer period. Artificial regeneration. The artificial regeneration is resorted on a large scale throughout India except in temperate regions. The artificial regeneration involves the following: raising of nursery stock. Nursery techniques. The nursery technique is varied from place to place and time to time. The nursery technique under traditional method of planting is raising of primary seedlings sown in the raised primary beds and transplanted into polythene bags filled with soil and maintained for 6 months. The grown-up seedlings are planted in the pits, duly removing the polythene bag. The nursery technique under the advanced technology is the clonal plants developed are vegetative cuttings, propagules, rooted under controlled atmospheric conditions and maintained for 6 months are used for planting. For this, the mist chamber and the clonal multiplication areas are established for the production of clonal plants. Earlier, CMA, that is 3% disco cornmeal agar, were used for obtaining the propagules and transfer to the tubes. Sandbed technology was developed to get the propagules from the mist chamber site itself to get quick propagules from the apical shoots of clones planted in the sand beds. Site preparation. Traditional method. Under traditional method, the site is cleared of miscellaneous growth and burnt. The planting points are marked by fixing the stakes at fixed escapement. The escapement followed earlier was 2 by 2 meter or 3 into 1.5 meter or 3 into 2 meter, etc. These methods were followed up to 1986. Thereafter, the site was uprooted manually and interplowed the area after planting. This method was followed up to 1994. Intensive method. From 1994 onwards, The government has taken up large-scale clonal eucalyptus plantations under the scheme aided by the World Bank. As per the program, the site is cleared of all the stumps through semi-mechanical uprootal using proclainers and the tillings the soil to 45 cm depth. Thereafter, the site is levelled and ploughed the area with heavy-duty tractors once before planting and once after planting. Cultivation techniques of casuarinas. Casuarinas are a very versatile group of plants with wide-ranging adaptability grow in different environments and provide multiple end users and services. They fix atmospheric nitrogen through a symbiotic association with bacteria Frankia. Casuarina wood with a high calorific value is a renowned fuel wood in the tropics. In India, Casuarina equisetifolia was introduced during the 19th century and is now estimated to be under cultivation in around 500,000 hectares, mainly in the peninsular region. Apart from fuel, the wood is extensively used for paper making, and of late it is a preferred choice for biomass-based power generation. The straight cylindrical stems find use in rural house building and as scaffolds in construction sites. It is the principal species for developing shelter belts in coastal areas and windbreaks for protecting agricultural crops. It also plays a key role in reclaiming mined areas and afforesting nutrient-poor sites. Casuarina seeds are small, consisting 5 to 6 lakh seeds per kg, but about half of them may actually be immature seeds which usually do not germinate. Germination is generally around 30% and about 30,000 to 1 lakh seedlings are obtained from a kg of seed depending upon source of seed and nursery efficiency. Seeds are sown in raised sand beds called mother beds of the size 10 into 1 meter. Generally, no pretreatment is necessary for casuarina seeds. In each bed, about 250 grams of seeds. In case of Casuarina jungiana, 50 grams are evenly spread by mixing with fine sand. They are overlaid with a thin layer of sand. The sand bed is covered with rice straw to prevent washing of the seedlings while watering. Cultivation techniques for Melia dubia. In Kannada, Hebbevu or Kadubevu. In Tamil, Malaivambu. Site factor. The trees grow well in sandy loam, red and laterite soils with an annual rainfall of 800 mm and above. Seed collection, processing and nursery techniques. Seed processing and pre-treatments. Germination is very poor. The drupes should be graded in water to remove floating seeds prior to sowing. Nursery. Seed sowing. It is best to sow seeds during March to April. Cleaned and dried seeds should be sown in the open raised nursery beds in drilled lines 5 cm apart. Seeds do not germinate in sand. They have to be sown in soil. Farmyard manure media in the ratio of 2:1 or 1:1 ratio can also be adopted. About 6 to 7 kg of dried drupes containing about 1,500 numbers are required for one standard nursery bed. The seeds sown need to be watered regularly, twice a day. Germination occurs within 90 days. Vegetative propagation. Juvenile stem cuttings and coppice shoots respond well to 1000 to 2000 ppm IBA in liquid formulation. Coppice and other trees respond better to rooting. Pencil-thick cuttings need to be taken for propagation. Thin shoots are easily susceptible to root rot. Drier seasons are conducive for rooting. Cultivation techniques for Melina arborea. It is also called Goomer teak or Shivani. Melina arborea is propagated by both seeds and vegetative methods. Seeds are sowed directly in the root trainer or raised beds of 0.6 meter in height and convenient length. The bed should be filled with sand up to a height of 0.5 meter. The seeds are closely placed in the bed in a row with a gap of 5 centimeters. The beds are to be watered twice in a day with the help of rose cans. 1 kg of Melina seeds contain about 1500 to 2000 seeds. The number of the seeds varies between the trees. Seeds normally germinate quickly and at high levels. Often the germination will be above 100% or more than one seed will germinate from each stone. The optimal temperature for germination is about 30 degrees Celsius and low temperature will reduce germination. The seed bed should be exposed to full sunlight as partly or full shade will reduce germination. After germination, the seedlings can be transplanted to containers. Summary: India is one of the 10 most forest-rich countries of the world. Recent forest management approaches have typically favored production of even-aged single-species stands. Intensive forestry practices and a willingness on the part of many forest owners and society to embrace a wider set of management objectives helped in going for alternative silvicultural approaches. They are characterized by a set of fundamental principles including avoidance of clear cutting, an emphasis on structural diversity and small-scale variability, development of mixed species with natural regeneration and avoidance of intensive site preparation methods. Many potential silvicultural treatments might be used to change, accelerate change or maintain tree and stand conditions. Those that are typically used to foster improved tree growing conditions and or improved growth and yield. The combination of treatments used in a silvicultural system can have large impact on growth and future yields. In this chapter, some of the methods for propagation of selective forestry species are described. Thank you.